In this video series, we are going to be covering the concepts of chapter 6 and 7, which has to do with protein behaviors. We are going to be focusing on enzymes, specifically what they are, their mechanisms, and how they are controlled. First, we need to discuss what are enzymes. When it comes to the composition of an enzyme, understand that enzymes are protein catalysts. And as we learned previously, a catalyst is going to be a molecule, in this case, a biomolecule, that is going to lower the activation energy for a reaction. Now, understand that proteins are not the only type of biocatalyst that we have. There are other type of catalysts that are called RNA catalysts or ribozymes, but we're not going to be focusing on them in these discussions. Now, understand that in biological system, the majority of biochemical reactions are catalyzed by protein enzymes. And when it comes to how enzymes are going to be acting on the different reactants, or like we call them in biochemistry, substrates, there are thousands of enzymes, and they are going to catalyze very specific reactions. Now, when it comes to the structure of an enzyme, this is going to be composed of the 20 amino acids that we have discussed previously that are the basic unit for proteins. Now, when it comes to the schematic model for an enzyme, as you can see, we have on the right side of the slide, we see that the enzyme is going to have a body, which is the area that is in yellow, and it's going to have an active site. Now, understand that the active site is where the chemistry or the chemical reactions occur. Meaning that's where we are going to be converting substrate to product. Understand that when we look overall at the structure of an enzyme, they are globular proteins. And when it comes to each enzyme, they have a unique tertiary structure that recognizes and binds a group of small molecules. Now, remember that the main goal of these enzymes are going to be the conversion of a substrate. And substrate is what we use in biochemistry, but remember that a substrate is another word for reactant. And that substrate that is going to bind to the active site is what gets converted to product. Now, how do enzymes work specifically? Understand that enzymes overall increase the reaction rates of the reaction, okay, by lowering the activation energy of the reaction. So they basically are going to be stabilizing the transition state. They do not change the standard free energy of the reaction overall. So let's look at two illustrations to see how enzymes work. And they're going to say the same message. So whichever illustration works for your brain to understand how an enzyme works, please select that works for you better. So let's understand that we have a particular reactant, okay, in other words, a substrate, that needs to be converted to products specifically. Let's just assume that is converting one reactant into two products. As we can look in this image, without an enzyme, those reactants have to climb, right, a tall wall. Remember that tall wall here is what we know as the activation energy. Just to remind you, activation energy is the energy barrier that reactants must overcome to be converted to product. Now, understand that if a particular reaction without an enzyme is going to have that tall activation energy in order to get to the product, if you run the same reaction with an enzyme instead, notice that now the wall that needs to be overcome to get into product is going to be shorter. Because remember, enzymes lower the activation energy. So, as you can see in the second illustration, that second illustration shows that if the overall goal of those reactions is to get to the product side, then sometime when the enzyme is added, what that happens to do with the reaction is that it lowers the barrier. So now we're going to be forming product in a faster rate because that energy barrier is going to be lowered. Now, let's look at a specific example of how this is. Here we have the reaction in which we have hydrogen peroxide being converted to water and molecular oxygen. This is a decomposition reaction. My shorthand for reaction is RXN. So, understand that if this decomposition of hydrogen peroxide, okay, is occurring and we have a relative rate, okay, it will have a relative rate of 1. I'm just going to focus on the values of the activation energy, specifically in kilojoules per mole. So as you can see, the height, okay, of specifically that wall from converting specifically hydrogen peroxide to water and molecular oxygen is going to be 75.2 kilojoules per mole. Now, if we use a platinum surface, which is going to be a chemical catalyst, notice that now the activation energy has lowered compared to 75.2. Now, if we specifically look at the rate of the reaction, is faster than the one with no catalyst. Let's now look at what happens when we have an enzyme. So catalase is an enzyme that catalyzes the decomposition of hydrogen peroxide. Overall, we observe that that energy barrier is even lower and the relative rate of the reaction is even higher. So in this example, we can observe that a chemical catalyst, yes, it makes the relative rate of the reaction increase, the activation energy of the reaction is decreased, but we can see that in an enzymatic reaction, meaning we're running the same reaction, but now we have an enzyme as a catalyst, is going to do the job in a more faster way because as you can see, it has a lower activation energy and the relative rate is faster compared to the chemical catalyst. Now, whenever we talk about the rate of the reaction, how fast things are happening, we need to talk about the kinetics. But also we need to take into account what are the thermodynamics of the system. So understand that the term thermodynamics talks about the heat that it is released in the system, okay? and how a reaction can be spontaneous or not. While the kinetics of the system tells you how fast, okay, or talks about the rate of the conversion from reactants to product. Now, let's understand. If we have a process that it is spontaneous, then that is said to be thermodynamically favored, okay? Because it is energetically favored, okay? What does that mean? If something is spontaneous, then there's no energy input that needs to be placed in the system in order to get to product. Now, when it comes to spontaneity, understand that it doesn't tell us anything about the rate. Let's give a specific example. We know that diamonds and graphite are going to be two forms of pure carbon, the element carbon. Okay, so they're two pure forms of carbon. Now, diamonds spontaneously turn into graphite. That is a spontaneous reaction. Energetically, it is favorable. Now, why is it that the diamonds that you may have seen somewhere do not turn into graphite right away? That's because the rate of the conversion from diamond to graphite is very, very slow. Whenever we talk about reaction progress, we need to review what is an energy diagram. And remember that an energy diagram or a reaction progress diagram or reaction coordinate diagram, I should say, just illustrates what is happening overall in the reaction. We plot the progress of the reaction in the x-axis against the free energy of the reaction. Okay? Now, you can see here specifically this diagrams is specifically for an exothermic process. And I know that this is exothermic because the free energy of the products is lower than the one of the reactants. Now, one of the main things that you need to know is what is the free energy of activation or deactivation energy. And as you can see, this is going to be the difference in energy between reactants and the first transition state of the reaction. Okay, remember, this is the energy barrier that needs to be overcome in order for the reactants to go to products. Sometimes when labeled, the free energy of activation is labeled as delta G naught double dagger. Okay, now in this reaction diagram, we also have delta G naught, which is the free energy change. And there's going to be the difference in energy between products and reactants. In some instances, this is also referred as the enthalpy of the reaction. I'm just illustrating how this relates to other terms that you may have heard in other chemistry courses. Now, understand that maybe you have heard of the terms exothermic, just like I wrote in the bottom, or endothermic. When it comes to biochemistry, we utilize terms that are very similar to those. So if you have something that is an exothermic process in biochemistry, we call it an exergonic reaction. And understand that when we look at the delta G naught for that specifically exergonic reaction is going to be less than 0. Okay? Remember, exergonic reactions tend to be as spontaneous because no external stimulus is required. We can also have endergonic process, which are going to be processes that have a delta G naught that is going to be higher than 0. And those type of reactions, we assume, okay, based on the conditions, they are not going to be as spontaneous because they are going to require an external stimulus.